Hello, welcome to the next in the series of review clips designed to assist you in preparing for your Biology 301 public examination. In this clip, we'll be taking a look at karyotyping. Simply put, a karyotype is a picture that shows the size and arrangement of chromosomes inside the cell of an organism. Chromosomes are arranged into pairs based upon their size and banding patterns, and this banding pattern comes from the unique sequence of genes within each chromosome. Typically, normally, there are 22 identical pairs of chromosomes in a karyotype and one set of dissimilar chromosomes. These dissimilar chromosomes are the sex chromosomes X and Y. Karyotypes can be used to diagnose genetic disorders such as trisomy 18 and a whole host of others. Karyotyping was established in the 1950s, realizing that the normal chromosome number in humans is 46. Remember, as previously stated, a diploid set contains 22 pairs of autosomes plus one pair of sex chromosomes. When the maternal and paternal set joins together, you get 23 pairs of chromosomes, with the diploid state being re-established upon fertilization. In short, a karyotype is an ordered display of an individual's chromosomes. It is a collection of chromosomes from mitotic cells. And when you have staining applied, this can reveal band patterns, it can reveal gross anomalies, which can be indicative of various disorders. This picture you see in front of you here now is of an actual karyotype that's actually done. The way things proceed in a karyotype, what happens is that the original cell with its nucleus is fixed during mitosis. Once the cell is fixed, a picture is then taken of the cell with the chromosomes being visible. These pictures, this chromosome picture, is then the chromosomes are cut out individually and they are paired according to banding pattern and chromosome size. This is then used to generate the karyotype. Once the original picture is taken, as you see here, it is then moved around, paired up, to generate what you see here in front of you. Each chromosome is paired up according to similar size and banding pattern for each of the 22 pairs, the 22 pairs of autosomes. Each of these chromosome pairs, which I'm placing squares around, represent what should be there, typically should be there, if the karyotype is quote-unquote normal. You also have, on the last set, a set of dissimilar chromosomes, which represent the sex chromosomes. In this case, because the individual is XX, what we have here is a female. In this case, going one step further, again, the chromosomes have a picture taken of them, fixed, and they are grouped according to banding pattern and size, cut out, and if everything operates according to plan, you should have 22 pairs of chromosomes plus the last two. In this case, because we have an X and a Y chromosome, this karyotype is of a human male. Sometimes the karyotype does not turn out normally. In this case, it is evident that you have 3 21st chromosomes. With trisomy 21, this indicates Down syndrome. In this case, because we all see XX, we know this is a Down syndrome female. I mentioned before about the banding pattern in chromosomes. This is done because the chromosomes are stained. They are stained with trypsin gymsma, and this staining method reveals a series of alternating light and dark bands that are turned to be G bands. Each chromosome arm is defined further by numbering the bands. The higher the number, the further that area is away from the centromere. Again, allowing you to group and place the chromosomes together as they should be. So again, the procedure is initially a sample is drawn and cells are cultured. They're allowed to divide. Remember, the chromosomes are condensed and they're visible during the stage of mitosis. This could be white blood cells. It could be amniotic cells. Again, samples from the fetal tissue. The chromosomes are fixed, mitosis inhibited by using drugs. Remember, usually they pair up along the metaphasic plate prior to being pulled apart by the spindle fibers. Drugs, colchicine or vincristine, inhibit spindle fibers, so the chromosomes then remain at the metaphasic plate. So they're fixed in place. Then, using a picture, the chromosomes are separated, the cells are burst, lysed, which causes the cells to separate from each other. They're then stained, a picture is taken, and then the chromosomes are cut out and arranged, paired up and arranged according to convention, size and banding pattern. Further technology which can be done are flow care, which involves a laser sorter. But the end result is the same. Chromosomes are paired according to banding pattern and size. So what's the benefit of karyotyping? Firstly, gender determination can be done. If it's XX, it's a female. XY is a male. Down syndrome and other genetic disorders can be detected if the karyotype shows a third 21st chromosome. Remember, the presence of this extra chromosome here in 21 indicates that the child has Down syndrome. Klinefelter's, another genetic disorder, XXY. Again, this karyotype reveals 2 X's plus the Y. Turner syndrome, another example. It has an X chromosome, but there was no Y chromosome. Again, this karyotype indicates the individual has Turner syndrome. Examinations have asked questions on this topic as well. For example, I present the following. Which genetic disorder could be diagnosed from the human karyotype below? If you look through here and analyze the karyotype, you will notice that the autosomes are all paired as they should be. There are no errors in the autosomes. However, when you get to the last set, the sex chromosomes, you'll note there's a single chromosome. This person has not an X chromosome, but no Y. This situation is indicative, this situation is characteristic of Turner syndrome, choice D. In this question here, you were asked, which chromosome abnormality is shown in the karyotype below? In this karyotype, you can see the autosomes are all normal and as they should be. However, when you get to the sex chromosomes, in this case the individual has X, X, Y. X, X, Y is characteristic of Klinefelter syndrome, choice C. The correct answer in this case is indeed that, choice C, Klinefelters. And this question asks, which is used to study chromosomes? Your choices are genotype, karyotype, pedigree chart, or Punnett square. The Punnett square makes direct reference to genetic patterns, which would reveal the genotype. A pedigree chart does trace the transmission of traits in generation and family histories, but the correct choice is choice D, karyotype. Thank you for your time. I hope you found this learning clip to be helpful.